intro to animals, which is pretty straightforward, I feel like. Mostly because we're all interested in animals because we are animals, right? So a lot of the stuff we talk about in this chapter and going forward will be much more familiar than things that we've talked about with, say, I don't know, single-celled eukaryotes or fungi or even plants, right? So we know a lot about how animals do things because we know a lot about ourselves. There's been a tremendous amount of research done into animals because, again, that's how we learn more about us, right? So you're looking for like model systems to test things like medical procedures or compounds in, you use other animals because they're the closest relatives to humans, since we're nested right in there in that group. So hopefully things become a little bit more familiar. Here will probably be still some things you guys have never heard of before, and that's what the class is for. So we'll learn some new stuff. And then after we get through, there are 4 animal chapters. Intro to diversity. So we'll talk about, this whole chapter is like characteristics of animals, basically. Then we'll do invertebrates and vertebrates. They have their own 2 chapters, kind of like plants, they're split in the middle, even though they belong in the same monophyletic group. And then there's 1 chapter on physiology, so we'll cover that after we get through diversity. And then we'll do a body systems overview. Okay, so that's going to be pretty human-centric when we start talking about body systems, but we'll throw in some other groups for sort of a comparative survey type thing. There are like 11 chapters on body systems, but we're going to just pick and choose bits and pieces from each. So I will have like 1 or 2 slides for each body system. So it's not going to be a chapter, it's going to be like a little taste, okay? We may spend a little bit of extra time talking about immune, just because it's interesting and relevant. So we'll see how much time we have when we get to that point. And then after that, that gets us up to exam 3. And then for the last quarter or so of the semester, we'll do ecology, which is usually pretty fun. People like ecology, and we're already doing stuff because we're all working outside on research projects. So we're already starting to talk about populations and communities and ecosystems. So it's kind of like, I think it gets easier from here, and usually more engaging because it's, you know, stuff that we're familiar with. So that's what we'll do today. We'll see how far we get, and then we'll decide when it's time to call it. Some of you guys are heading out to the preserve after lecture today. Some of you are not, so we'll just kind of go from there. If we end a little bit early, I'm okay with that today, since you guys have to take a test. So, you ready to start some animal stuff? Who likes animals? Fair. All right, there's lots of diversity. These are just, again, some cool pictures to get us started. So this is just, you know, aesthetics. Nothing in there, no information. All right, so we're on to Kingdom Animalia. Oops, sorry. Earliest animals evolved in the oceans over 600 million years ago during the late Precambrian. So before, this is the earliest animals, not the earliest land animals. Okay, so first they were in the water, just like plants. So we'll talk about those earliest groups. We'll start with the oldest lineages and work our way forward to the youngest lineage that we'll talk about. And then we'll along the way, we'll talk about that transition to life on land from water, okay? So that's coming up in future chapters. Here's our same theme that we touch on every time we start a new group. How many are there and how many do we think there are, right? The same pattern holds true every time. We think there are about 1.5 million described and living. Okay, so that 1.4 million number doesn't include extinct species, which there are plenty of those. But there's an estimated total number somewhere between 3 million and 30 million. That's a pretty big range, right? So again, it's just that same idea that here's what we've described, here's what we have labeled and taxonomically classified, but there's a whole lot out there that we don't know, right? So the same theme, you guys, again, don't have to memorize those numbers, just kind of be familiar with the idea that there's always more to learn and discover than what we already know. Classification system of animals, we're going to spend quite a bit of time talking about these characteristics that we use to classify animals, including anatomy, evolutionary history, embryonic development, and genetics. So nothing new. Same way we classify other organisms. The one thing that's going to stand out a little bit in this chapter is the embryonic development stuff, because we haven't really talked about that too much in any other groups. Right? So we'll spend a little bit more time looking at that in animals. Animals share a huge variety of body forms, right? So think about something like, a jelly, or a worm, or a bird, or you, right? So tons of diversity, right? Varying levels of complexity. Compare a sponge to an ape. Yes? Again, that sort of huge variety. We'll talk about organization. So there are lots of ways to organize body plans, but there are plenty of characteristics that all animals share in common. So that's where we'll start. Same as our other chapters, following that same pattern. All right, the first bullet here should be easy. All animals are eukaryotes. We already know that because, first of all, we're animals and we're eukaryotes, right? But I told you guys after we did bacteria and archaea, everybody going forward is eukaryotes. So there's just one chapter on prokaryotes and everyone else has that true nucleus and those other characteristics. All animals are multicellular. Remember, there's that one sister clade of protists that falls right next to animals, the choanoflagellates. Because if you are to fall into animals, you must be multicellular. Okay, so that's something that all animals share in common. Most animals have specialized tissues, so we'll spend some time talking about the types of tissues that you find in animals. And tissues are just groups of cells that cooperate. Okay, they're specialized cells that work together to perform a particular function. So it's that looking at that evolution not only of multicellularity, but also complexity of form, which leads to function. So that's what we're going to spend some time talking about over the next several chapters, is that idea. So we'll look at those tissue types. Animals are all modal. What did we say motility refers to? If you're modal, what can you do? You can move by yourself, right? What does it mean to be mobile? You can be moved. Somebody can plop you into a wheelbarrow and push you around, right? But that doesn't mean you're moving on your own. So all animals have the ability to move from place to place, at least during certain life stages. So there are going to be examples of sessile animal species. What does sessile mean? Not moving, right? So rooted in the substrate, not going from place to place. But even animals that spend part of their life rooted in place have been able to move at some point. Okay, so maybe there's a larval stage that swims, and then as that larval stage matures, it finds a spot and some roots down there. So we'll see some stuff like that. But at least at some life stage, all animals have the ability to move under their own power from one place to another. All animals are heterotrophs. So what does that mean? We all eat stuff. Yeah, none of us can photosynthesize, no matter how hard we try. Huge variety of strategies even within the heterotrophic nutrition system in animals: carnivores, herbivores, omnivores, parasites, all of them. What do omnivores eat? Plants and animals, right? Pretty much anything. What about herbivores? Just plants. What about carnivores? Just meat, yep. And then parasites live inside of a host or on a host and get their nutrition that way. So we'll see that sort of variety there. Most animals reproduce sexually. There are exceptions. It is the exception rather than the rule in animals, but there definitely are examples, and we'll look at a few examples of that, asexual reproduction in animals, but mostly you see the haploid, diploid sort of reproductive cycle. Life cycle is described as diplontic. So this right here, you guys, is the reason that I talked about alternation of generations in plants at all, okay, because it's contrasting. So remember, alternation of generations means that you have two life stages, and both of those life stages are multicellular, but one is haploid and one is diploid. You guys remember that? We just talked about it on Monday. That is called being haplodiplontic. I didn't give you guys that term. I don't care if you know it, but we are considered diplontic, which means that our major life stage is diploid. So the only time we have a haploid stage is in the gametes, and those are single cell, a sperm cell or an egg cell, and that's it. As soon as they merge, as soon as you have fertilization, that first cell, that first zygote is diploid. It's mitosis from there on out, right? So you are diplontic because the majority of your life cycle is diploid, and you have that in common with all of your animal relatives, okay? And we talked about this last bullet a little bit as well, with plants being the sort of foil, right, or the comparison. We talked about plants having indeterminate growth, meaning they can put on new tissue throughout their life, right? You can cut off limbs and new limbs will grow, leaves fall off, leaves grow again. Animals are different than that. We have a predetermined body plan written in your DNA that determines your morphology. So what you look like as an adult organism is set, right, by your genetics. It's to follow a developmental pathway, this step and that step and that step. And at the end of the day, that's what you look like. And then if you lose a part, you likely do not grow it back. Do you think there are exceptions to that, the regeneration thing? Yeah, anybody know of any animals that can do that? Starfish can do it. Say again? Some salamanders, axolotls are those Mexican ones, you know what I'm talking about? They are a big model organism in research for regeneration biology. They can do that too. But most animals can't regenerate. But all animals follow the determined body plan. So those are your features. This is also really nice because it's a neat little group. Okay, so these characteristics, it's not like, well, sometimes it's this, sometimes it's that. Sometimes it's multicellular, sometimes it's single cell. It's all pretty straightforward. There are exceptions. Most have specialized tissues. We'll look at a group that doesn't. Okay, but for the vast majority, you're going to see those specialized tissues. Most animals reproduce sexually. There are exceptions, but again, remember I said those are the exceptions. So most of these characteristics are a little bit kind of more set, right? You see it more frequently. You can kind of count on those to be the case a little bit more in animals than you can in other groups. So that's kind of nice. Questions about these features of animals? Makes sense. It's all stuff we've talked about. It's just specific to animals now, right? Okay. All right, so we'll spend a little bit more time going into some detail on each of these bullets that we've covered. So we're going first for tissues. Okay, so if you're not familiar with what tissues are, again, this is a little bit more in-depth for you on that. So a tissue is a collection of cells that had a common embryonic origin. Okay, we're going to talk about this again in a minute, so don't panic too much if you don't know what I'm talking about. Cells come from the same germ layer. So we're going to look at germ layers as we look at animal development, and I'll give you a little bit more detail, sort of tie it back into tissue formation. But what we're looking at basically is that the cells that are going to end up as part of a tissue, whether it's muscle tissue or nervous tissue or connective tissue or epithelial tissue, they all sort of start out in the same place in the developing embryo. So you think about development, you start out as a diploid zygote. How many cells is the zygote? One, right? It's the first diploid cell, and then mitosis, right? So 1 becomes 2, 2 becomes 4, so on and so forth. So we'll look at a few of those developmental stages, but as that little ball of cells is forming, you start to see specialization of those cells. So if you just had a zygote that just went through mitosis and every cell was identical, by the time you became a fully formed human with however many billions of cells you have, if they were all identical, you would just be a big blob of cells. Big blob of identical cells, right? But that's not the case. We have eyeball cells and liver cells and white blood cells, neurons.
Is it the ecto, mesoderm?
Yeah, endoderm, mesoderm, ectoderm. Yep, that's exactly what we're talking about. So we'll take a look at some of those in schematics in a few slides, and we'll talk more in detail about what those are. But it's just a layer of those early developmental cells as they start taking on their specialized function. All the cells that are going to become, let's say, muscle, come from the same place in that developing embryo. Does that make sense? So we're talking about common embryonic origin. We're talking about those layers, those germ layers. Okay. Again, we'll get some more information on that later. Cells in a tissue are grouped together to perform a specialized function. So they don't operate in a vacuum. They're connected to other cells that are also specialized for that same function. So if you're looking at something like muscle tissue, it's made up of a whole bunch of different myocytes that are muscle cells that are communicating with each other, that are cooperating with each other, that are connected to each other, working together to perform the same function. So you've got specialization and you've got cooperation, okay, in cells that make up tissues. There are shared morphological features. Well, that makes sense. If you are specialized as a cell to perform a job with other cells that are also specialized to do the same thing, you're all going to be shaped the same way. And you're going to be shaped in such a way that allows you to carry out your specific function efficiently. Okay, so that's what we're talking about there with shared morphological features. And the same thing with organization. So they're lined up, they're connected, they're communicating, they're organized in a way that helps them to, again, pull off that function that they're specialized for. Your 4 main tissue types, you guys don't have to go into a ton of depth about this. There's way more information in your textbook than what I expect you to know. All I want you to be able to tell me is what is the function of each of these 4 main tissue types, and recognize examples, okay? But just the ones that are given to you, so no trick questions. You've got nervous tissue, muscle, connective, and epithelial tissues. Every animal does not have all 4 of these, okay? But if some animals do, you have all 4, right? So if you're looking for tissue, you're looking for one of these 4 types, or somewhere between 1 and 4 of those types. Makes sense? Hopefully that's clear what I'm trying to say. All right, so the function of nervous tissue. What does nervous tissue do? You can look at the slide, or you can just tell me what you think it does. What do your nerves do? Yeah, communication, right? Transferring information throughout your body. Your body parts are integrated and know what the other ones are doing through your central nervous system, which is your brain, right? So your brain, your spinal cord, and all of your peripheral nerves are all made up of nervous tissue, nerve cells, which are neurons. So I want you to know that the functional cells of nervous tissue are neurons. That's these guys. So these little, they're pink in this picture, and they have all these processes coming off of the cell body. So you've got information coming in to these structures, which are called dendrites, and then you've got information leaving out through the axon. So they're communicating with each other in a chain to make sure that you know what's going on externally, internally, and everything in between. Okay? So the whole plan with nervous tissue is to transmit nerve impulses, and those nerve impulses are information. Yeah. It's almost completely neurons. Yeah, there's going to, well, any nervous tissue is going to be, all the functional cells are neurons, and then there are tons and tons of support cells, but they're tiny compared to the neuron. So if you were to take a slide, like a slice of brain tissue and make a slide and look at it, you'd be able to see, it kind of looks like this picture. So you see all these interworking, interlocked neurons, but all that yellow stuff is cells called neuroglia or glial cells, and they're support cells. They do things like insulate, provide nutrition, active immune cells. So there's other stuff in there, but all the function is going to be neurons. So your brain is just a huge tangle of those neurons that support cells. All right, makes sense? Nervous tissue, information superhighway. Okay. Muscle tissue, what does it do? You guys know what muscles do, right? What could you not do if you didn't have them? Move, right? So muscles contract to produce movement. Now you're going to see different types of muscles. Skeletal muscle attached to your skeleton. That's why it's called skeletal muscle, right? And those are voluntary, and they contract so that you can move around, or so that you can stand still. And it also takes muscle contraction to maintain posture, not just flop over into a pile. But all muscles are going to contract. So you can have locomotion as in skeletal muscle, or you can also have movement within the body. So you have muscles that you can't control, like the smooth muscles inside of your digestive tract. They still contract. You're not choosing to do it, but it's happening outside of your conscious control, involuntary contraction, but it's still causing movement. It's causing movement that pushes food through that canal. Make sense? You have smooth muscle that line your blood vessels. Okay, they control blood pressure. Still contracting to produce movement. So sometimes it's about transport. Sometimes it's about homeostasis. Sometimes it's locomotion. Cardiac muscle contracts to produce movement. What kind of movement? Cardiac muscle, do you guys know? Yeah, heartbeat. So that's anytime you see muscle is contracting, the cells always get shorter to provide force for motion to happen. And it's always causing some kind of movement, whether it's voluntary or involuntary. Connective tissue is structural support and transport. So you're looking at bone and cartilage, so your skeleton and skeletal elements. Adipose is another example. Does anybody know what adipose is? Fat. Yep. So your fat tissue and blood is considered a connective tissue. Even though it's not structural, it's transport, right? So you've got fluid connective tissue and structural connective tissue. Those are pretty straightforward. And then finally, epithelial tissue, which is all of your linings and coverings. Animals are extraordinarily well packed. Okay, so all of your organs are lined and relined, and all of your organ cavities are covered with linings so that things can move smoothly, so that things stay hydrated, so that things stay protected. So you've got epithelial tissue lining and covering everything. So internal and external linings, the best example that you can see is your skin. Your epidermis, the top layer of your skin, is epithelial tissue. And it's just a particularly tough and dry version of it. So most of your epithelial tissues, like inside your nose or lining your stomach, are going to be moist. Your skin is not, usually. Right? You have to actually put lotion and stuff on to keep it from drying out. It's there to protect you. But that's an external example of epithelial tissue. Okay? So this is it for tissue types. This is okay? Most of this is probably familiar, at least to some extent, right? All right, so let's talk about the true tissues thing. What the heck does that mean? Most animals have them, so most animals exhibit one or more of those 4 types of tissue, except sponges. Sponges are still animals. They're still on the animal trees. We'll take a look at this here in just a second, but they don't have true tissues. They are still multicellular. They have cells that do specific jobs. So there's that specialization within that multicellularity, but the cells don't necessarily connect and communicate and cooperate with each other. They might do it, but they don't have to. So in other words, you could take a sponge, drop it in a blender, turn it on, whip it up into a sponge smoothie, and those cells will still continue to do the job they were doing before. So they don't rely on one another to do those same functions. So some of the cells are specialized for filter feeding. Some of them are specialized for transport. So they do their specialized jobs, but they don't have to do it together. So they're not true tissue. So we look at the animal phylogenetic tree. Here's our root. This is your last common ancestor of all animals, right? The population that would have been here. So this is called metazoa, all the animals, right? So when you see zoo, you think animals, right? Like you go to the zoo, zoology, that's animal stuff. So metazoa is all animals. And if you look down here, this branch, look here, it's all by itself. What is the term for a branch that looks like this? Early branching, not a whole lot of diversity. What kind of lineage would you describe that as? Reading a phylogenetic tree. Nobody wants to take a stab at it? We would say that that is the most basal lineage, right, of animals. So there's debate about what the most basal lineage of animals is, and we'll talk about that later. But for now, this tree that I want to present you with is sort of the standard phylogeny that we're going to work with, at least to start off with. And that puts sponges here, basal to and sister to all the rest of the animals. So sponges are called parazoans. Para meaning beside or next to, okay? And then the other branch, look at this label. Everybody else here in the rest of the animal tree are the eumetazoans. What does that EU prefix mean, you guys? True. So you've got metazoa, which is animals, then you've got eumetazoa, which are the true animals, so to speak, and you've got parazoa, which is beside the animals, or beside the rest of the animals. Okay, so you got sponges on its own branch, the parazoans, then you got eumetazoans, that's everybody else. The difference here is no tissues versus specialized tissues. Okay, so when we say most animals have the tissues, it's everybody except for these dudes, okay, except for the sponges. So why are they still called animals? Well, again, they're multicellular and those cells do specific functions. They're also heterotrophs, so they meet the other criteria to be considered an animal. Okay, so they fit, they're just weirdos. Okay, essentially a really old lineage. Okay, not the first animal, but a really old basal lineage of animals. Good? All right, let's talk about reproduction and development a little bit here. As we mentioned on that first slide, most animals are capable of sexual reproduction. Most animals use sexual reproduction. And for many, it's the only option. Okay, so we said there are exceptions, asexual reproduction, we'll look at some of those. But for most animals, we're talking about the whole process of fertilization. So you got a small motile male sperm cell. Again, what's motile mean? It moves, yeah. Travels to a larger sessile female egg. So what is sessile again? Doesn't move. So the female egg is usually larger. There's a lot more stuff in there than is contained inside of a sperm cell in most cases. So the sperm cell is smaller and it moves. So it travels to the female egg and fertilization occurs. That is the combination of the male and the female gametes. You guys probably all learned that in like middle school, yes? Birds and the bees type stuff. Okay, birds and bees do weird stuff, but I'm just talking about fertilization. Other behaviors too, but nonetheless. Okay, so you're looking at combination of gametes. You're talking about the fusion of your gamete nuclei to form a zygote. So first, the cell membranes fuse. When sperm approaches egg, the membranes fuse first. And then you have karyogamy, which is the fusion of the nucleus. Okay, so once you get that nuclear fusion, you have a haploid nucleus in the sperm cell and a haploid nucleus in the egg cell. What does that mean about those nuclei, the chromosomes in the nuclei, if they're haploid? How many sets of chromosomes are in each one? One. Yeah, haploid means you got one set of chromosomes. So if you got one set in the sperm cell coming from the male parent, you got one set in the female egg cell coming from the female parent, they merge, they fuse those nuclei, and two haploid nuclei give you a diploid nucleus. First diploid nucleus, that's your zygote. Okay? All these pieces are falling into place, yes? Okay. All right, so fertilization can happen internally or externally. All right, internal fertilization is most frequently seen in animals that live terrestrially. Okay, think about keeping your gametes from drying out. Okay, protection from desiccation. You can't just leave your gametes lying around and hope for the best on land, right? But in the water, you kind of can, right? So you see things like some fish that just lay eggs in the water, and then the male comes along and just deposits sperm on top of them, and fertilization happens externally. Okay, so you see examples of both, and usually it's land versus water. Okay, again, there are a few exceptions to that, but that's a good general rule. So once fertilization happens, you get cell division through mitosis, yes, because zygotes are diploid, so diploid cells divide and make more diploid cells. Those cells start to differentiate, meaning they start to take on their different shapes, roles, functions. Okay, so differentiation occurs. You follow a genetically determined developmental pathway. The cells become more and more and more specialized until ultimately you get specialized tissues. You end up with that determined morphology and physiology. So what your body form looks like and how that form leads to your ability to do certain functions. Okay, so form and function are like this, you guys, tightly married. Okay, this whole chapter on animal physiology, chapter 30, I think it is, no, 33. Doesn't matter. The fourth animal chapter we'll go over all about form and function, okay, and how the two are intimately related. So we're always talking about both. Young animals may resemble adult animals. They may look just like their parents, only small, okay? Or they can undergo metamorphosis, which is another term you guys have probably heard before. What is metamorphosis? Yeah, drastic change, right? So give me an example. You can look at the board if you want to, of an animal that undergoes metamorphosis in its life. Butterfly, yeah. What does it start out as? Well, an egg is laid, right? And the egg hatches and you get a caterpillar, some kind of larval stage. Then that larva is going to pupate. It's either going to become a chrysalis or it's going to build a cocoon, depending upon the type you're looking at. And then a few weeks later, out hatches a butterfly. Does this dude look anything like this guy? Not really, not morphologically, and not physiologically either. Right? There's a similarity in maybe color pattern. We'll talk about that later when we look at learning coloration in the ecology section. But form-wise, these look like two completely different animals. Right? What about this one? What's that? You guys don't know what this is. We need to have frogs, right? You guys know tadpoles turn into frogs, yes. I don't mean to be flip, but you probably know this. Yeah, so tadpoles also come from eggs. If you've seen frog eggs before, they're just like little clear balls with a little black dot in the middle, like a whole bunch of eyeballs stuck together. They hatch into tadpoles. What do tadpoles lack that frogs have? Legs, yeah. So they go through a series of developmental stages where they grow out those legs, and then this guy can leave the water. Amphibian, right? Two ways of living, water, terrestrial. Yes? Okay, so that's metamorphosis. What about something like a human? Do you go through metamorphosis or do you look like a tiny adult when you're born? Tiny adult. The dimensions, proportions are a little different, right? Huge head, little body, but you pretty much look like a small human. You just have to grow into yourself. So those are the differences between young looking like the adult or undergoing metamorphosis, okay? Just some terms to get you started thinking about that. All right, as promised, we'll talk about a couple of examples of asexual reproduction. because it does happen, even though sexual reproduction is for sure the preferred method and the only method for many types of animals. You see asexual reproduction in lots of invertebrates, so things that do not have one. Sorry, nope, I'm talking about invertebrates. What do invertebrates lack? Do you guys know that? Vertebrae, yeah, spinal cord. Yep. So anything that doesn't have a backbone. So things like insects, octopus, slugs, what else? Starfish. Any kind of animal that doesn't have a backbone is an invertebrate. We'll cover all of those groups in the next chapter. Some fish can use asexual reproduction. You see it in some amphibians and even in some reptiles. So that just means you don't have to do fertilization. So there are some weird ways of doing things out there. However, to this point, we've never seen asexual reproduction in birds, and we've never seen it in mammals. Okay, so those are the exceptions to the exception, I guess I should say. So you'll never see asexual reproduction in those two groups. 3 examples. Okay, you guys just want to know a definition of these, okay, for the test, which is, exam 3, so far from now. Budding is when you actually grow like a bud. So this is a hydra. A hydra is in the same group of animals as like a jelly. Okay, so they have stinging cells usually, things like that. They're sessile as adults. They usually produce larvae. So they have a swimming larval stage, so there's that motility piece, but they can also do budding. Okay, what that basically means is, look, it just starts growing a little bud off the side of its body, and then the bud develops the needed parts, tentacles, mouth, and then pops off. Just make yourself a little clone. Okay, what do you think these guys look like genetically? They're identical. Yeah, it's a clone. So there's no genetic diversity in budding. But there are benefits to being able to do this. What do you guys think is the benefit here? Can you imagine what that might be? When would it be helpful to be able to do this? When you're getting old, and can a hydra move? Can't really go anywhere, right? It's sessile at this point in its life stage. So if it hasn't been able to sexually reproduce, maybe just bud, right? Cut your losses and just grow a clone. Okay. Fragmentation is similar to budding, but also different. So we're looking at a starfish here. Fragmentation is when you lose a piece and that piece regrows another entire individual. Okay, so it's not like growing an individual that then separates. It's losing a piece and then that piece regrows into a full individual. That makes sense. So they're similar but different. Some starfish do this on purpose. So they'll actually like inject the leg, and the leg will grow into a new individual, and the old starfish regrows the leg. So we talked about regeneration, right? That's possible. Sometimes it happens accidentally, like a starfish is accidentally fragmented by a predator or caught in a fishing net or who knows what, and it breaks accidentally and then regenerates. Okay, some species are really good at it. Other species you have to have like a big enough piece, like just the tip of the foot won't regenerate a whole starfish, but some can. So that's fragmentation. You guys see the difference between the two processes. All right, and then parthenogenesis, that's its own whole thing. We won't really go into a ton of detail about it, but what you just need to know, parthenogenesis means literally virgin creation. Okay, so there's no fertilization that happens. A completely unfertilized female-born egg develops into a whole new individual. Okay, sometimes it is done for sex determination. So like in bees, a diploid female bee will make eggs. They don't get fertilized, and those become male bees. So you get your drones. Here you got a diploid female bee, lays some eggs. They get fertilized. Wait, this one is what I'm looking at here. Diploid female bee makes diploid eggs, or a haploid male bee makes sperm. You get diploid female bees. Okay, so you don't have to have fertilization, but if you do, it can end up changing the gender or sex of the bee offspring. So all you really need to know, you don't need to know these complicated patterns, just know what parthenogenesis means. Okay, sometimes this is what you see sometimes in reptiles, which is really kind of weird. There are certain species of lizards that will do this just because there's no male lizards around. Well, gotta make offspring somehow, so just have one. It's pretty weird, right? Usually when that happens, they're all female. So interesting stuff. All right, cool. You guys doing okay? All right, let's see. I think this will be the last thing we talk about, so we'll stop a little bit early. Okay, so you guys can go get ready for your test or get ready to work on your lab stuff and then take your test or whatever. So let's do this, and then I'll tell you why we're gonna stop where we stop after we get through this. So embryonic development, this is similar across animals. You guys may remember way back in the beginning of the semester when we talked about using embryonic development or embryology as evidence for evolution. You can compare pathways and compare stages and look at the way that animals or other organisms develop and use that those shared pathways as evidence of relatedness. Right, so this is a really good example of that because all animals are going to do a series of developmental steps that are going to look just like this. All right, at least to some extent. So now if you're not that complicated of an animal, you'll stop at certain points along the way. But a lot of these cellular processes that happen in embryonic development are shared across animals. So we're going to learn a few of the steps. Okay, there's a lot more to it than this. If you're heading into anatomy and physiology, when you get to the second half of that, you'll spend a lot of time talking about embryonic development. So good luck. The zygote undergoes cleavage. That's just the term for splitting in half, right? So you get that sort of line that forms, and then the zygote splits into two cells. So that's what cleavage is, a series of mitotic cell divisions. The cell divisions are mitotic, right? It's mitosis. So the zygote is what, diploid or haploid? Diploid. So the resulting cells from this mitotic division are also, yes, diploid to diploid. So mitosis is going on. 3 divisions later, so zygote becomes 2, 2 becomes 4, 4 becomes 8. We call this the, you guessed it, 8-cell stage. That's pretty straightforward. Why do we care about the 8-cell stage? Because at this point in time is when cellular differentiation actually starts. So it only takes 3 divisions from the zygote before you start to see these cells becoming different from each other. They're not yet becoming sealed in their fate, okay? But the earliest evidence of differentiation is starting to appear. What happens is you start to get polarity, okay? So you start to get a top and a bottom. Instead of just having a cell with a nucleus that's kind of an amorphous blob, they start to take on differences in like what's on each side of the cell. So the earliest evidence of differentiation in cells happens at the 8-cell stage, which is why we sort of stop and look at this blob of 8 cells. Because cool stuff is starting to happen already there. The next stage is the blastula, and the blastula is a hollow ball of cells. Okay, so this ball is solid as it's starting to divide, and then between the 8-cell stage and the blastula stage, it becomes like a sphere with a hollow center. It's not empty; it's full of fluid, but it's hollow. Make room for that fluid. You guys with me? So you got the blastula, which is a hollow ball of cells. This is important because what happens next is called gastrulation. So this is always tricky, blastula versus gastrula. Okay, so that's where we're headed. Gastrulation is the process of the blastula becoming the gastrula. All right. This is not just me being mean to you and making you learn terminology that's hard to remember. This is actually where you see the formation of those germ tissue layers that are going to become specialized tissues later. Okay. So this is another sort of stop along the way that's really important. So what happens is that blastula continues, cell division is ongoing, it continues to divide. It starts to fold in on itself. You guys see this right here? This little infolding of the cells. So basically you've got this ball of cells and it starts to just dip in a little bit. It starts to just fold inward onto itself, forming multiple cell layers. And once you have those multiple cell layers, you've got a gastrula. Okay? Gastrulation is when these germ layers form. So the blastopore, that is this hole that forms where the wall of the blastula folds in to start gastrulation. That is the blastopore. That hole is forming the future digestive system. So early, while you're still just a ball of cells, your digestive system starts to form. In an animal that has a full digestive tube with a hole at both ends, You guys follow me, right? Mouth, anus, okay? A tube through the middle, that blastophore is gonna continue all the way through till it opens up the other side. And that will become the digestive tube. So that's really important, a really important phase in development. Also, what's happening is you're getting those germ tissues. So like Doug was mentioning, they're called endoderm, mesoderm, and ectoderm, and we'll get to this in more detail later. But you've got this innermost cell wall that's starting to form is going to be your endoderm. Okay, it's inside. Then you've got ectoderm on the outside. And if you have an animal that's complicated enough to have that third layer, it's mesoderm and it forms in the middle. So you start to get these layers. And those layers are specialized. So endoderm is going to eventually give rise to specialized tissues that end up in the inside, like the lining of your lungs. Mesoderm is going to give rise to tissues that will become connective tissue, like blood or muscle or bone. Okay? And then your ectoderm is going to give rise to cells, tissue types that are going to become neurons and skin, stuff that's kind of on the outside. Okay? So that specialization of cells, the way that your cells are actually starting to migrate in this little ball of cells is really, really important, even not early on in embryonic development. Okay? And all animals are going to do this to some extent. So you get gastrulation, you get germ layers forming, which is going to be that origin of those embryonic origins of those tissue types. And then the last term I want you to know is organogenesis. If genesis means creation or beginning, what do you think organogenesis means? Formation of what? Organs, yeah. So organs come later, all right? Because organs are made of, guess what? Tissues. So you got cells that start to differentiate and cooperate with one another, become different tissue types. And then later, you've got different tissue types that sort of layer up and start to cooperate and become organs, kidneys, intestines, heart, etc. Okay? All right, that's a good place to stop. We're a little early, but you guys deserve it. We've been through a lot already of chapters, and we're sort of starting this, as you know, before the exam, that we'll cover this, well before the exam. We're going to pick up with Hox genes on Monday. The reason I'm stopping here is because this gets a little bit hairy, right? So I feel like it's a lot to throw into your brain today, and then expect you to remember it later because we're preparing for that test. So that's a good reason to stop there. So we'll pick up talking about Hox genes. We've got a pretty good video that will help you through this. I usually do a supplemental instruction session before exam 3 on this topic because it gets a little bit, like I said, kind of muddy. So we'll talk about Hox genes, how they form, what they do, and what happens in gene duplication events. So it's getting a little intense, which is why it's a good place to stop. Then we'll go into how we classify animals, and that'll be the second half of this chapter. Okay, so next week.